Today we are talking about the genetic code. So the genetic code is a linear arrangement of the nitrogen base in the mRNA, that is the messenger RNA. Although the linear arrangement of nitrogen bases in DNA is said to determine the sequence of amino acid indirectly, and there are only 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. It means the precise sequence of these 4 nitrogenous bases on the DNA strand somehow direct the proper amino acid to their proper places through the mRNA. That's why the genetic code actually present on the mRNA. So many theories are proposed to explain the mechanism by which a particular sequence of nitrogenous base in DNA, rather than well accepted theory which proposed by the F.A.C. Crick. The theory holds the existence of a genetic code and its smallest unit, which codes for one amino acid, one amino acid, is known as codon. So codon is the arrangement of nucleotide sequence in the mRNA which codes for a particular amino acid. Whereas the genetic code is the sequence of nitrogenous base in mRNA molecule which encloses information for the synthesis of protein. Actually, this one is the appropriate, that is, it encloses the information for the synthesis of protein, that is the synthesis of amino acid chain. So come to the what are the characteristic features. Let's come to the genetic code actual number of genetic code. First of all, there are total number of 20 amino acid. So for the coding of these 20 amino acid, the combination of only one nitrogen base or 2 nitrogen bases cannot provide sufficient code words. But if we assume that only one nitrogen base can code for amino acid, then there are only 4 codons. Again, if the 2 nitrogen bases code for amino acid, then there are 16 codons. So that these 4 and 16 codons still insufficient for 20 amino acid. So the three possibility of the 3 nitrogen bases can code the 20 amino acid, although the 3 nitrogen bases will give 64 codon, which are more than enough to code for 20 amino acid. It is evident that several of the triplets have the same letters but in different sequence, and these code for different amino acid. So the genetic code are in the 3-letter code, and that's why it is known as triplet in nature. And this triplet in nature can code for the specific amino acid, and this also decided by the process of in vitro synthesis. Scientists have found out codons for all the 20 amino acid, and these 20 amino acid we have already know, and we can also find in any reference book or textbook of this amino acid, and also the genetic codons. So genetic codons are triplet in nature, and these triplet in nature or this triplet genetic code can codes for amino acid, and this genetic code contains 64 codons. Out of these, 3 are stop codon or known as termination codon, and 1 initiation codon. So out of 64 genetic code, 61 is functional and rest 3 are the termination codon. According to Crick, only 32 types of tRNA could be sufficient to transport 20 amino acid. Okay, because of this nature of nitrogen base, one tRNA can recognize several different codons coding for the same amino acid. Okay. So one tRNA can recognize several different codons for the same amino acid. Let's come to the characteristics of the genetic code. First one, genetic code is a triplet. The genetic code that specifies the amino acid in a polypeptide chain comprises of a sequence of 3 nitrogen bases on mRNA. 3 nitrogen bases. That's why this is known as triplet. As for example, AUG is a genetic code, CUU is a genetic code, GUU is a genetic code. So these are the triplet in nature. Next, genetic code is commaless. The nitrogen bases which are present in mRNA are read continuously without any internal punctuation. Without any internal punctuation. It means there is no comma in between the adjacent codon, and each codon is immediately followed by the next codon. Each codon is immediately followed by the next codon with no intervening spaces. As for example, the genetic codon CGA, CGG, CTC, TTT, CTTC, TAC, AA, AAG. So there is no internal punctuation is present. Okay. Next, come to the third character. Genetic code is non-overlapping. The codon in mRNA are read in successive groups of 3 nucleotides in the direction of 5' to 3'. Since the successive group of these nucleotides there is no comma, so that the nitrogen bases are read out in a non-overlapping form. That is, translation of genetic code in an mRNA begins at the correct point and there is no overlapping of the codons. That is, the successive presence of the genetic codon does not overlap with each other. All are the unique, or all are the different from one another. Next, come to the fourth point. Genetic code is degenerate in nature. Degenerate. If we look that there are 64 possible codons, out of which 61 is functional and codes for 20 amino acids that contribute to protein. These multiple system of coding represent degeneracy system. As for example, the serine amino acid is coded by UCG, UCC, UCA, UCG, etc. The valine is coded by GUU, GUC, GUA, GUG. Okay, this is the degeneracy of genetic code. That is, one amino acid is coded by more than one genetic code. Okay. Next, genetic code is unambiguous. Very, very important. We have told that the genetic code is degeneracy in nature, that each one amino acid is regulated or coded by the different genetic code, but a specific genetic code can only codes for a specific amino acid. Okay, a specific genetic code can only codes for specific amino acid. That is, one amino acid can be coded by the several genetic code, but one genetic code can only code for a single specific amino acid. So this is the unambiguous. Okay. Next, come to the fifth point. Genetic code is almost universal. The same genetic code is said to be present in all kinds of living organisms, including virus, bacteria, unicellular, multicellular, etc., excepting some genetic code. Rather than all the genetic code is said to be present in living organisms. Next, these are the characteristic features, and the start codon is AUG and the stop codon is UAG, UAA, UAG, UAG, UAA, UGA. Okay, these three stop codon, that is UAA, is known as ochre, UAG known as amber, and UGA is known as opal. Ochre, amber, opal. Okay. Next, if we analyze the degeneracy of the genetic code, then it can be shown that a amino acid, that is suppose serine, is coded by the UCG, UCG, UCC, UCA. In these three genetic code, the first and second nitrogen bases, that is U and C, are fixed, whereas the third base becomes variable, GCA, UCG, UCC, UCA. So the variability or versatility or flexibility of these third base is known as Weibull base, and this hypothesis is also known as Weibull hypothesis. This Weibull hypothesis was proposed by Crick in 1966. According to this hypothesis, the base in first position of anticodon the base in first position of anticodon on tRNA is usually an abnormal base. Okay, and this base is known as Weibull base or fluctuating base, and this base can be paired with any nitrogen bases. Okay, so this is known as Weibull base, and the hypothesis is known as Weibull hypothesis, and this Weibull hypothesis is present behind the degeneracy nature. So genetic code can also start the protein synthesis, and genetic code can also stop the protein synthesis. Now I talk about two scientists. First one is the Nierenberg and second one is the Khurana. Marshall Warren Nierenberg, an American biochemist and also geneticist, successfully synthesized protein in cell-free protein synthesizing system obtained from the E. coli. This was described as cracking of genetic code. So Nierenberg responsible for the cracking of the genetic code. It showed that UUU coded for phenylalanine. For this discovery, he shared 1968 Nobel Prize along with the Hargobind Khorana. So second scientist, Hargobind Khorana. Scientist Khorana was born in 1922 in Raipur, Chhattisgarh. He made remarkable contribution in artificial synthesis of nucleic acid. He discovered how to synthesize triplet RNA molecule of known sequence, thereby assigning the genetic code. He also synthesized artificial genes, and Hargobind Khurana also shared the Nobel Prize in 1968 with Marshall Warren Nierenberg and Robert W. Foley. So, in genetic code synthesis and their characterization, both the scientists have played a great role and also awarded Nobel Prize in 1968. So this is the genetic code and these are the characteristic features. So there are question is this: what is genetic code? What are the characteristic features of genetic code? Write a short note on Weibull hypothesis. Give a short explanation about the cracking of genetic code or contribution of genetic Khurana, contribution of Hargobind Khurana, Indian biochemist, in the genetic code characterization or synthesis, and what are the stop codons, what is the start codon. These are the some questions for UG level. Okay, thank you.